Pues muy buenas tardes a todas y a todos. Nos da gusto verlos el día de hoy en este seminario con el que inicia el semestre, los seminarios del Doctorado en Desarrollo. Good afternoon, Dr. Lindemann. It's very nice to have you here at CEPROVI. It's a pleasure that you're opening our seminars for this semester. And it will be really interesting to hear your lecture. Pues muy bienvenidos a todas y a todos. Yo deseo que es un semestre de mucho provecho en cuanto a estas oportunidades que tenemos de compartir con expertos en los diferentes temas asociados a las líneas de investigación. También, por supuesto, agradecer a los organizadores por estas actividades de este semestre. Sin más, pues, welcome again, everybody, and we can now open this seminar. Thank you again, Dr. Lindemann, for being with us this day. Muchas gracias, Dr. Arturo.
Gracias, Gaby. Thank you. I will introduce to Dr. Stephen Lindemann. Dr. Lindemann is a bachelor in microbiology with honors in the molecular biology, biochemistry, and genetics of Purdue University in 2004. In 2010, is PhD in microbiology in University of Iowa, Carver College of Medicine. After the doctoral preparation, Dr. Lindemann has a postdoc position in between 2011 and 2013, and after senior research scientist in the microbiology group, Biological Science Division in Pacific Northwest National Laboratory in Richland, Washington. After is a adjunct professor in the Jean and Linda Bioland School of Chemical Engineering and Bioengineering in Washington State University in Pullman. In 2016, is assistant professor in the Department of Food Science at Purdue University. At present, is associate professor in the same Department of Food Science. Is a professor also in the Department of Biological Science in Purdue University as well. Dr. Lindemann has around 57 peer-reviewed papers in journals of high impact in the area. Is invited talk in different associations and companies in USA. Oral conference also in different conferences in US. Dr. Lindemann has a research support for the investigation that is carried out in this moment. Is principal investigator, is a grant from the U.S. National Science Foundation Chemical Bioengineering Environmental and Transport System Division. Is around 2.4 million of American dollars. In 2099, is principal investigator, also with a grant for U.S. National Institute of General Medical Science, with a budget around 1.9 million of American dollars. Dr. Lindemann participates in different societies as Institute for Food Technologies, American Chemical Society, American Society for Nutrition, International Society for Microbiology Ecology, and American Society for Microbiology. Really, it's a privilege to have you as speaker today. It's a privilege that you open this seminars, this cycle of seminar in our doctoral program here in the CEPROVI. Thank you very much. I appreciate it.
Thank you, Luis, for the very, very kind introduction. It's a pleasure to talk to the students today and tell you a little bit about what we are doing with understanding the relationship between cereal bran fibers and how the microbiome responds. And so I was a little ambitious with my title today. After I started actually building the talk, it became obvious I can't talk about genotype and processing today. So I'm going to focus on processing, and maybe some other time in the future, we'll talk about genotype more. So to start, here's the outline for my talk. I'm going to start by introducing carbohydrates and how they interact in the colonic ecosystem. I'm then going to move to understanding how we work on bran particle sizes and how we've seen those influence the microbiome. And then we'll start specifically talking about how milling method influences how particle sizes and interacts with the microbiota. And then we'll end. So let's start with the first part. This is my favorite graphic to describe kind of what happens in the gut microbiome. And this is the path of a water molecule over about 100 hours in time. And so the reason I like this graph is because it shows perturbations that are varying both in amplitude and frequency. So you have waves move the particle back and forth just a little bit, low amplitude, but high frequency. You have tidal movements that every 6 hours, in and out, larger amplitude, intermediate frequency, and then you have also some trajectory of an entire current-based system. And the thing that I like this is these are kind of the day-to-day dietary perturbations. We have dietary pattern perturbations where we change what we eat. We have larger influences on our gut microbiome, and then the waves are against that larger change. And then we live a whole life, and we start where we're born and we end when we're older. And then this period of time is directional as well. So the gut microbiome is both malleable and also stable. And what that means is that individuals' microbiomes can interact differently in idiosyncratic ways, individual ways with the foods that are being consumed. And so one area of interest to us is sustaining diversity in the gut microbiome. And that is because the loss of diversity seems to be linked to many kind of chronic Western diseases that we want to try to avoid. And further experiments with diversity, here is a mouse experiment performed in the Sonnenberg lab at Stanford and described by Eric Martens in this paper, in this graphic. Mice are fed, for example, high fiber diets over generations, and they're allowed to transmit their microbiota from mother to pup, and they do a pretty good job of maintaining the diversity of microbiota on the high fiber diet. But if they are raised on a low fiber diet, the efficiency by which they transfer microbes is much reduced. And so further, the farther they go, the less recovery you get if you switch them back to the high fiber diet, suggesting that these organisms can actually locally go extinct in some microbiomes. So we are interested in making sure that we provide an environment that's supportive of diversity. The way in which we measure diversity in the lab is using this molecule here. This is the 16S ribosomal RNA. It is essentially the ribonuclear protein scaffold for the bacterial ribosome. And because of that, there are regions of this molecule that are really important to its overall structure. Those can't evolve very well or very quickly because they have a lot of selective pressure against them to maintain the overall structure. But there are variable regions inside that touch the nuclear proteins or the protein sides of the ribosome. And those are free to evolve alongside their proteins. So what this allows us to do is it allows us to design PCR primers against the conserved regions and amplify using those conserved regions. And at the intervening in the middle, we have DNA that is representative of that variable region, which is telling us where that microbe comes from on the tree of life. So the primers being conserved allow us to target nearly universally bacterial and archaeal organisms, and the intervening spaces serve as a name tag of which organism that sequence came from. The problem is that the polysaccharides and consumption of polysaccharides by different microbes varies at very fine taxonomic levels. So sometimes at the species, sometimes even at the subspecies level. So here you see a set of gut microbiota. They are related organisms, Eubacterium rectali and Roseburia inulinivorans and other species of Roseburia. And here is their performance and growth on a variety of different carbon sources. One is fructooligosacchariide. 2 is galactooligosacchariide. 3 is xyloligosacchariide. 4 is amylopectin, arabinan, beta-glucan, arabinoxylan, arabinogalactan, and inulin. And what you can see here is that even among the 3 strains of the species Eubacterium rectali, they vary in their ability to consume these carbon sources. So more growth is more yellow. For example, this M104/1 species is not able to consume arabinoxylans at all, where its near neighbor organisms can. None of the species of inulinivorans, Roseburia and inulinivorans can, but species of intestinalis are able to. But their performance is not equivalent. So this is a very finely variant trait in microbes. So the problem is compounded by the fact that we start with really, really different microbiota amongst people. And so here are different phylum level abundances. So big, large categories of microbes and then family level abundances of microbes here. The purpose is not to have you stare at these names and understand these patterns, but just to get an appreciation for how different these people are. And you might think these are people from different parts of the world or with very different dietary patterns. The reality is all of these people are from the upper Midwest in the United States, the states of Wisconsin and Minnesota. And they are very, very different in the structure that they present with. To give you some context for that, these here are all members of phylum Chordata, all the animals here with backbones, obviously from very different ecosystems. They have very different roles in their ecosystem. You would learn very little about an ecosystem, for example, if all you did was quantify how many chordates were there. These are the members of family Ursidae. They are the members of the bear family. Obviously, they're much more similar than the organisms here, but they all have different temperature preferences, different food sources, different ways of interacting with their environment. And so even knowing how many overall bears there are wouldn't tell you necessarily whether you were in the polar north or whether you were in a bamboo forest in China. And so therefore we have a very hard time understanding how microbes, predicting microbial function from these kinds of analyses at high levels. And further, there's a complicated microbiota profile, but there's also, and in response to, a complicated glycan feeding profile for what we're actually providing our microbes to eat. Most of what we're talking about or thinking about are fibers here in the space of plant polysaccharides, but then also there are microbial glycans produced, there are dairy-derived oligosaccharides, and residues from mammalian tissue that we don't process very well or can't degrade ourselves, like sulfated polysaccharides. But additionally, there's an endogenous source of microbial available glycans, and that is our mucus. The idea currently is that if we lack these other categories of dietary glycans, microbes may turn on consuming, and we have evidence that they do turn on consuming the glycans we produce for the sugars they need to survive, but that then reduces the thickness and the ability of our mucus to separate our tissues from the microbes inside our guts. However, we talked about diverse glycans that are available. It turns out also that unlike humans, microbes are pretty rational consumers, which means that they compete strongly for the carbon sources that give them the most energy for the least investment of their own energy in producing the proteins they need to consume them. And so what that means is we set up longitudinal gradients from within the colon. So there's very, very intense competition for soluble glycans that are easy for microbes to ferment. And then as we move through the ascending and transverse colon and then into the descending colon, we switch to colonization of particles and things that are hard and require lots of energy and enzymes for microbes to ferment. And so we have this longitudinal gradient of what is being consumed where, and very different, we think, communities active in the ascending versus the descending colon. The end product of fermentation of fibers is almost exclusively at the community level, the short chain fatty acids. And the short chain fatty acids are important to us because they control lots of human physiology. So gut-derived short chain fatty acids, acetate, propionate, and butyrate influence things like how our liver processes glucose, whether it generates more glucose, whether it stores glucose in lipids, or whether or not it consumes those fatty acids to make energy. And overall modulates our lipid buffering capacity and total metabolism. Also, it modulates our immune systems so that, for example, when an antigen from an organism, here is Bacteroides fragilis polysaccharide A, is detected by a dendritic cell, which is an immune sentry cell, the fate of this interaction determines on how much butyrate, propionate, and acetate are present in the system. So that the responses of the dendritic cell, and then also the T cells that might see the antigen presented by the dendritic cell, are responsive to whether or not there's a lot of short chain fatty acids, and determine whether this T cell will respond in a way that promotes inflammation or inhibits inflammation. Here is the T regulatory cell might be formed. So there's two ways we can think of to improve the output of short chain fatty acids in the gut. One is so if we're here in this environment right now, most of us in the West have a low diversity microbiota, and we have fairly high diversity of carbohydrates, but the metabolic output isn't so high because the overall diversity of microbiota is fairly low. One can change that in two ways. Either you reduce the fiber complexity, meaning you only feed the fiber that the microbes can consume. And that here is the bottom strategy, where we only feed this black spiral carbohydrate to the green microbe, and we produce whatever the green microbe's metabolic output is, which here is propionate. Or we feed, we increase microbial diversity. And if we have increasingly diverse microbes, we have microbes that are able to degrade all of these carbohydrates, and they can produce overall higher production of short chain fatty acids and diversity of short chain fatty acids. So I don't know how the data look for the Mexican population, but I can tell you that Americans are very, very bad at eating their fiber, which you probably have observed, at least in seeing what our dietary habits are. This is a significant problem. So we are receiving approximately 40 to 50% of the required or recommended dietary fiber every day. And what that means is there's substantial challenge for underfeeding our gut microbiota. And so my lab is interested in understanding what parts of fiber influence function. And I've put fiber there in quotes because, as many of you may have seen a nutrition facts panel, this one or a very similar one, where dietary fiber is a single line here. Sometimes, if we're lucky, that line is split into two. It's soluble and insoluble fibers. And still, these categories encompass all plant polysaccharides that humans are not able to degrade. So there's a huge, huge category of molecular structures present there. And so if this is all the resolution you have in measuring, and this is all the resolution available to the consumer, this is the dietary advice you're given, which is, it doesn't really matter what type of fiber you eat, just you should be eating fiber, so choose ones that you find comfortable to consume. And this is intensely practical advice if all of these molecular structures perform the same way, but it's really perhaps not correct advice if these molecular structures perform differently in the gut microbiome. So what my laboratory is working on is trying to identify what fundamental properties of complex carbohydrates are altering how microbes respond to those carbohydrates. Our null hypothesis is that there are no generalities in fiber microbiota interactions. That means that all behavior, it's idiosyncratic either to the fiber structures themselves or to the genomes of the organisms that are fermenting them. But our hypothesis is that there are general ecological strategies that microbes are using to gain advantage with respect to certain fibers. And what that could be is how they hydrolyze polysaccharides or transport what they hydrolyze, how they regulate their genes, what their growth rate is and affinity for these carbohydrates. These are genome encoded properties which result in community level properties. If these generalities do exist, they provide us a foundation for engineering fibers and engineering foods, and potentially even breeding plants for predictable gut microbiome outcomes. So let's talk a little bit about where we started here with this project, which is on brand particle sizes. And one of our earliest observations was when we were trying to test two different methods for simulating passage through the upper gastrointestinal tract. Here, the destarching, so starch removal and protein removal of two methods, and we did that against a variety of different brands. And we found that the performance of those different methods was actually dependent upon which type of species we were looking at. All of them had increased reduction of starches from method A, but our second method only removed protein in a positive direction, removed more of the protein for wheat bran, and the rest of them were either no difference or were actually worse for doing that. I want to call your attention to the difference here specifically between sorghum and wheat brands in the case, because quantitatively, these brands were similar in terms of their overall digested content. So with method A, we have approximately 2% to 3% starch remaining, and we have approximately 14 to 15% protein remaining in these after we digest them with method A. But regardless of the fact that we had pretty similar overall macromolecular compositions, OTUs are different operational taxonomic units. OTU stands for operational taxonomic unit. It's a computational analog of a microbial species. And what that means is we can't prove that these are actually different species. All we have is a sequence, so we don't know if they're from the same species or not. But we draw some boundaries inside the sequences to say these sequences belong together. They cluster together. And therefore, we can quantitate how many of the sequences fall into each pile that sort of simulates what species should look like in terms of their abilities. What you can see is, though, there are very significant differences in how different organisms respond to different brand types. So sorghum, for example, Bacteroides ovatus is much more abundant here on sorghum bran than it is on wheat bran. And vice versa, the Prevotella are more abundant on the wheat brands than they are on the sorghum brand. So this idea here is that there are some specificities. So here's Prevotella, I forgot to note for you here. There are some specificities, it seems, for how certain organisms are responding to these specific brand types quite independently of the overall macromolecular composition. So we had the hypothesis that the Westernization of diet and its response to decreasing particle size may be important in modulating how microbes respond to particles of wheat bran. And that is when I talk about Westernization, we start with more traditional methods of processing foods, which then yield to intermediate methods like the stone mill in grinding foods. And then now we have the brutal efficiency of the roller mill to produce fractions and then specific size ranges of fractions as well. So as societies move along this axis, what is definitely true is that the average particle size of their food decreases. So in order to do this work at the time, an undergraduate in my laboratory, Ariana Romero Marcia, took some hard red spring wheat bran that was provided by Metal Milling Company. She sieved it out into different size ranges. And then we in vitro digested it as we talked about before, to remove starch and fat and protein. And also then after we performed in vitro digestion, we exposed it to microbiota from 3 individuals and allowed it to ferment. And then we evaluated the community of microbes that resulted through 16S sequencing. And then we measured the short-chain fatty acid production using gas chromatography. And what we saw very early was that there seemed to be gradients in responses of certain species to the brand types we provided. So here's the finest category. And here's the largest category after 48 hours of fermentation. What you can see in this case, more abundance is orange and less abundance here is blue. Some organisms seem to have preferences that responded to these various size fractions. And if we plotted those out on bar graphs, it becomes a little more obvious than the heat maps. You can see that, for example, this Bacteroides OTU is much more abundant in small particles than it was in large particles. And so we can see a lot of this kind of gradient behavior. And if we looked at the short chain fatty acids I didn't even include the short chain fatty acids. I'll include the short chain fatty acids for the second study. One of the drivers of this was the chemical composition of these particles. So what we found is that our smallest particles had much more glucose in them than the largest particles did, and there was a gradient of glucose. And so we thought, well, what we're seeing here is differences in overall starch availability here to microbes. And so therefore, there's an obvious gradient that's responsive to the starch. In order to control for that variable, Ariana then took the 1.7 millimeter Brand fraction, the largest sieved out. So she sieved out that size fraction. She then put that through the cyclone mill and re-sieved it out and generated a new size range of Brand fragments derived from this original parent Brand. And also broke this into two categories. So she broke the 300 to 180 micron category into 250 to 300 micron and 180 to 250 micron. And what we found is that very clearly she was able to eliminate the glucose effect. So all of the particles that were derived from this Brand had roughly the same, or at least statistically indistinguishable, amount of Brand fiber or of glucose in the Brand fiber. And it did reveal something else that was interesting though. So the arabinose to xylose ratio here, with the arabinose to xylose ratio, is an indicator of how branched the arabinoxylans are. And arabinoxylans are hemicellulosic polysaccharides that compose about 50% of the dry weight of brands. So a very, very common fiber in the structure of these brand cell walls. What she found is that the 180 to 250 micron category had the highest branch density of these arabinoxylan polysaccharides, whereas in the largest particles, we had much lower branch densities there. And so a postdoc in my laboratory, Eunice Tunchel, and a graduate student then at the time, Ria Thakur, both have since gone on to do amazing things. They took this project and then decided to find out whether or not it was influencing the community structure. So the question was, do we still see the same phenotype? Even though the glucose gradient that we had before is gone, do we still see the same microbial response? And so when it comes to short chain fatty acids, what they found is very similar to what we found the first time, is that there is an inverse correlation between the production of propionate and butyrate by the gut microbiome in a size-dependent way, meaning that we see the highest production of propionate from the smallest particles, and we see the highest production of butyrate from the largest particles, meaning that there seems to be some kind of trade-off here between production of these two molecules. And that correlated really strongly with the microbial population. So here we're looking at 6 species, operational taxonomic units of bacteria, and their abundance on various fractions. So we've colored this by the rainbow. So smallest particles here are purple and like short wavelength, and then the largest particles are red, so longest wavelength. And then open bars here are the bacteria that are associated with the supernatant end of the culture. And these hatched bars are those bacteria that are associated, the abundances associated with the different particles in those cultures, because of course these aren't soluble, so we have particles and separate and liquid we can separate. So you can see is that there are a variety of different strategies that microbes are employing. So this Bacteroides seems to prefer the supernatant of small particles. This here, Bacteroides, this OTU7, Bacteroides plebeius, definitely prefers small particles, but prefers to attach to them rather than be suspended in mix. And then vice versa, we see this Oscillobacter, for example, prefers larger particles than smaller ones. So we started to see some very significant differences in how microbes were colonizing and growing on these fibers. And I just wanted to demonstrate to you how diverse this can be even within really, really small groups of bacteria. So I'm going to show only the Bacteroides species here on this heat map. So this Bacteroides species prefers all supernatants, but especially large particles. This one prefers all supernatants, but especially small particles. This one here prefers smaller supernatants, but attaches to all particles. This one is particle associated with a preference for small particles. This one is supernatant associated with a preference for small particles. So these are all the different Bacteroides behave so differently in the context of this community. This is not something that's happening as a property, for example, of the Bacteroides, just like we were talking about as a property of the bears. This is a property that relates to this individual species. And so right now, I'm working with a project with Dr. Ash Fricker, who is an alumna of my lab, and she is working on processing metagenome sequences from these communities to determine what is it that makes certain microbes display these different patterns, what genes in the genome are related to that. So let's talk for a minute now about the milling method effects. So this is now work where we're transitioning to, all this is done by a graduate student in my lab, current graduate student in my lab, Miguel Alvarez Gonzalez. And he is following up on what has been done with the cyclone mill with different milling methods which are more relevant to industry. And so what we find here is that, you know, the cyclone mill is great for laboratory work, but not so replicative of what's happening out in industrial processes. So he took hard red winter wheat bran. He then sieved it out to obtain the largest size fraction, just like Ariana had done. And then he subjected it to milling with the micro hammer mill, and then a roller mill, and then our research stone mill here in the laboratory. And so generated the same ranges of particle sizes, sieving them out after milling, across all three methods of milling, but having generated those in different ways. And so here's the sieve, and from that we see our particle size ranges that result. We have what passes through the sieve, less than 180, then 180 to 250, 250 to 300, 300 to 500 microns, 500 microns to 850 microns. So he then digested those particles in the same way that he had before with using our method A of in vitro digestion. And so the first step here is in pepsin. Pepsin degradation is a broad spectrum approach that has enzymes like amylase, trypsin, lipases, ribonucleases, and proteases to try to eliminate all of these materials as much as we can and leave the fibers. And then also we have amyloglucosidase treatment, which removes the glucose from starch. And so what he then did was he took cultures that were sequential in nature, so that he took individual cultures and passaged them one to the next. And so what he had done then is engineered evolutionary bottlenecks between cultures, meaning that organisms have to be abundant in order to be transferred to the next culture. So over time, what happens is we dilute out the microbes that are not growing well, and we retain the microbes that do grow well on our substrates. So what this does then is select for the most efficient bran-consuming microbes. So he took fecal samples individually from 3 individual healthy donors. He's processed 2 of them so far, and I'll show you the results from those. He then performed fermentation in sodium phosphate buffer, which has some trace elements and some additional nutrients that we provide, we provide low concentrations of amino acids and vitamins. And then we allow the microbes to ferment these one to the next. And so the important part here is that these tubes form lineages. So this first tube goes to the second tube, goes to this third tube in lines. Same thing for the second tube, they don't cross ever. So we have independent observations of cultures that are developing on these bran fibers with time. And he analyzed them with 16S ribosomal amplicon sequencing, short-chain fatty acid analysis, performed analysis of the gas production and pH, and then also did SEM imaging of the residual particles over time. And I won't have time to show all of those things, but we'll show you mostly the 16S ribosomal RNA stuff. So the interesting thing is that the bran tissues specifically provide different resources for and inhibitors of different microbial consumption patterns and different microbes themselves. And so, you know, as you start here with the alurone most proximally to the endosperm, and then you move up through the inner pericarp and outer pericarp, the composition of these bran fiber layers actually changes pretty dramatically. And so, you know, where we see many more proteins found in the alurone layer, we find much of the way as xylans and beta-glucans and cellulose is found in the upper bran layers. And so Miguel was able to show how his milled cells, here's our outer pericarp, here is our intermediate layer here in the middle, and then at the base we have these larger alurone cells that you can see here as well, light microscope and electron microscope. And so Miguel is incredibly patient, and so he was able to physically dissect the brands using a dissecting microscope and get pure fractions of just the outer pericarp or the intermediate layer or the alurone cells at the base. And he was then able to get enough to determine what was the monosaccharide composition of these individual layers. And what he found was that, so here's our code right here. So arabinose is in gray and xylose is here in gold. What he found is that the outer pericarp arabinoxylans were much more branched than the inner pericarp, intermediate layer and the alurone layers. And further, the alurone layers seem to retain a substantial amount of glucose, even though there's little evidence that alurone is synthesizing starches. It's possible, especially with small granules of wheat starch, that disruption of the alurone layer may allow granules to nest inside these cells and be protected from some enzymatic degradation. So that's something we're currently working on. So you look at that arabinose to xylose ratio, as we talked about before, the outer pericarp is much more branched than the other two. And it's analogous to work that we had also done here in this paper, in another paper by Eunice Tunchel, where he was looking at the differences between arabinoxylans extracted between hard red wheats and soft red wheats, and found that there were subtle structural differences there. So makes us wonder whether or not what we're observing here has to do more with what fractions of the cells we've extracted rather than underlying genetic differences. But that's a different story for a different day. I'll happily answer questions on it if you're interested though. So the different milling methods that Miguel employed influenced the arabinoxylan content of different size fractions and had different curves for arabinose to xylose ratio. So interestingly enough, we see that the stone milled overall, we have higher, and significantly so in some cases. In some cases, there are no statistically significant differences, but stone mill in all fraction types had the highest amount of arabinose per unit xylose, and the roller mill had the least. You can see that if you look at the individual bar graphs here, you can see that different milling methods have statistically significant differences, especially between hammer and stone and roller mills. So when it comes to the microbes that are fermenting these cultures, interestingly enough, what we found here are the supernatant species richness. And so species richness simply means how many different species do we observe in the culture at different time points. So on day 1 here and day 7 from the supernatant cultures, and from the particles day 1 and day 7. And what you can see is that overall the number of organisms that we observe in the supernatant decreases, and the amount of organisms we observe in the particles increases over the sequential cultivation. And that makes sense. It means a number of these organisms here at the beginning really are not involved in bran fermentation, and they can't do anything with it. So we lose those species with sequential cultivation. However, we enrich for the species that are able to bind to particles and to consume those particles. And so interestingly, the supernatant microbiota that we observed were related to particle size, although not nearly as strongly as the particles. We'll talk about it in a little bit. So here is the composition of that first fermentation after one passage. And you can see that there are some minor differences among these milling methods, but it's really hard to see major differences in terms of the community structure on day one. There's a little bit of a trend here in donor 2, where in this case, we see this pink organism here, which is a Prevotella species. That Prevotella species seems to decline with increasing particle size. We've observed this before in other cultures as well, but donor 1, we didn't really see a whole lot of difference. By day 7, however, we get to a point where there are milling method associations with certain numbers of Bacteroides. For example, the relationships of these individual purple bars here, most of which are the Bacteroides species, the abundances of those are milling method dependent, and they're also size dependent, where we see, for example, more overall Bacteroides in smaller particles and less overall Bacteroides in larger particles. The relative abundances and differences of these are, in terms of their structures, are at least somewhat different in the supernamed. We see stronger effects on those in donor 2. So moderate effect in donor 1, stronger effect in donor 2, where we see increasing populations of the members of phylum Firmicutes. Generally speaking, depending on the milling method, those are different members of Firmicutes a little bit. There's variation in which ones are more abundant or less abundant. But more than that, we see reduction overall in the amount of Bacteroides and increases in the abundance of Prevotella on small particles. But these effects were fairly minor. Oops, graphics here. These effects are fairly minor, as you can see here. So yes, we see an effect, for example, on donor OTU1, donor Bacteroides ovatus. We see that OTU1 Bacteroides is much more prevalent in all types, but then especially we see that the responses of these two organisms are very different between donor 1 and donor 2. However, the particle-associated microbes show significantly different behavior. So in this case, here's donor 1 on day 1. This is donor 2 on day 1. We see much stronger effects in donor 2 right off the bat after our first fermentation. We see variation in the amount of organism Prevotella coprii, and we see a lot of difference in the Bacteroides population sizes, specifically here, this one particular Bacteroides species, or this is not a Bacteroides, the Firmicutes species here. Sorry for the misspeaking there. So these different Firmicutes species are differentially abundant based on particle size, where Prevotella in donor 2 is abundant, and then these members of Firmicutes, especially this OTU here, this which we'll talk about in just a minute, is much more abundant on larger particle sizes. And if you look at that after 7 days of cultivation, the effect is substantially larger. So specifically with Prevotella, so Prevotella species were much more promoted by our roller mill than they were by our hammer mill or our stone mill, for example. These Prevotella were much more abundant when we had roller milled in donor 1 microbiota. And then we saw the same thing was true for donor 2, except for the fact that in donor 2's microbiome, Prevotella was only really abundant in the smallest particle size fraction for any milling type, although it was much more abundant for the roller milled than it was for the others too here. So what you can see here is that, this is the elevated roller mill population of Prevotella here. And you can see that also in donor 1's microbiome. So this Lachnospiraceae species, so this is a member of Firmicutes, this is this member here. What you can see in the Lachnospiraceae is that we see also milling method dependent patterns of abundance for different milling methods here with respect to this species too. And so interestingly enough, this species seems to prefer larger particle sizes, and also its ability to colonize seems to be dependent somewhat on what milling method you employed to start with. So finally, the short chain fatty acid production that was observed for donor 1 and donor 2 are dependent on particle size as well. So in the first case, we saw what we had seen before, where we produced the opposite of what we'd seen before. We saw what we saw for propionate, which is in the first passage for these organisms, we saw a much higher production of butyrate than we did in larger particles, at least for the roller milled particles in both donor 1 and donor 2. The others are kind of flat with respect to particle size in terms of their butyrate production. So this is actually counter to what we've observed in the previous study, but it might have something to do with the fact that instead of using mixed microbiota, we're using individuals microbiota this time around. However, if you passage these cultures over sequential generations for 7 days, what we were able to do is select for populations that were more butyrogenic for the roller milled fractions at lower particle sizes. And we saw different responses then from the stone and roller milled, hammer milled in the middle in terms of their overall short chain fatty acid production. And so this is also true. We saw some patterns also of acetate formation. So in this case, we saw high acetate produced and small particles on day 1 for all organisms. And this may be a simple fraction. The reality of the set that the smallest particles might be more overall fermentable. But at the same time as that's true, we saw a very similar pattern develop with respect to acetate production in day 7 on donor 2's microbiome, but not with donor 1's microbiome. So if you go back to see what that looks like for donor 1, in butyrate, we saw differences here that were present in donor 1's microbiome and less present in donor 2's for butyrate. With respect to acetate, the pattern is reversed. So we see more strong patterns with day 7 acetate production in donor 2 than we do for donor 1. With respect to propionate, propionate shows us the same graph like we had seen before, which is that propionate production is especially high on early small particles early on and late in fermentation in the succession trajectory. And that was true for all milling methods, but especially true for roller milled produced. And you can see that relates, we think, here to the amount of Prevotella abundance, which is, Prevotella is a known propiogenic organism. And we think that the amount of propionate is directly responsive to how much Prevotella we have fermenting these brands. Likewise, you can see that the relationship of the Kinneothrix firmicutes is a putative. We think it's a butyrogenic organism, relates to the particle size. So the abundances relate to particle size here. We think that those two things are also related in that way. So finally, with respect to milling method and its induction of different ways for microbes to attach and consume these brands, Miguel took these different particles and he took them to the scanning electron microscope right after they'd been digested by in vitro digestion. And what he found was that the tissue breaks in different ways, as probably you expect and probably everybody expects. With respect to the different size ranges, though, the morphotypes are really different. So for example, our stone milled fragments, we have these fibers that are sticking up and flying off of particles and provide different attachment sites. Whereas intermediates and large particles maybe look more similar than the smaller particles do. And he also looked at colonized particles. And so what he did was then he in vitro digested them just like he had before. He inoculated them, isolated the particles from the inoculated cultures, and then looked to see where the microbes were attached. And what he found was that the attachment was almost exclusively here on the alurone side of the bran. You can see these are microbes decorating the surface here. We saw very, very little colonization of the pericarp. And that probably makes sense. Those pericarps are resistant to microbial degradation. That's their purpose that they serve in the field for protecting the grain. But they also then mean that the bottom side is likely to be where the area of attachment is for the microbes. And this is something that Miguel is currently following up. He's working on understanding the mechanisms of this and also processing his third donor's samples to try to identify what are the reasons why microbes are attaching to the side and converting it. How are they converting and metabolizing these cell wall structures? So finally, Things to think about: all bran particles that enter our diet are probably not the same with respect to how they interact with our microbiota. Specifically, the milling methods that we are using generate different resources for microbes, and they do so differently as a function of particle size, and that's probably because they have different compositions of tissues. Those milling methods, though, intermediate how microbiota colonize and metabolize the bran structures that we're feeding them. And so what that means is that if we're producing flours by stone milling versus roller milling versus hammer milling, we might be differentially feeding our microbiota in ways that are potentially interesting for health. And finally, we may be able to mill and mix the bran particles that we're consuming and generate soluble fibers from them for targeted impacts on the gut microbiome with respect to its short chain fatty acids and its population sizes of different microbial species and strains and genotypes. And so with that, I'd like to thank you for your attention. I'd like to thank Miguel and Eunice and Ria, Ash Fricker and Dr. Fricker and Ariana Marcia for doing the work. You've met them in the course of the presentation. I also like to thank Bruce Hamaker and Brad Roos because they taught me how to speak carbohydrate chemistry. I'm a microbiologist by training, so this was incredibly helpful. The USDA funded this work with respect to the brands, and I'd also like to acknowledge the Army Research Office, the National Institute of General Medical Sciences, and then the National Science Foundation for their funding of the lab. And with that, I'd like to take any questions you have.
Okay, thank you very much for your excellent talk, Steve. I appreciate. In this moment, we open the session of everyone has a question. Please, you can open the microphone, or if you want, you can to attend to use the chat for the question, and Ariana support the translation. Up to you. I don't know, Javier, if you want, you can ask directly.
Yeah, I see one question.
Or you, okay.
The bacterial total count in the microbiota. So are you talking about inside the gut, or are you talking about inside the tubes? Javier? He's probably typing.
Sorry, I mean, I said, the question was in the gut.
Yes, so that depends on where you are. In the stomach, it's very, very low, so approximately 100 to 1,000 cells per mil. But in the small intestine, it grows rapidly, depending on up to, at the top of the small intestine, maybe it's about the same, 100 to 1,000 cells per mil. But in the terminal ileum, it can be in the 10 to the 7th to 10 to the 9th range. In the context of the colon, there can be 10 to the 10th to 10 to the 12th cells per gram of contents. So a huge microbial population in the colon.
And how about the predominance?
Yeah, so that depends
different
greatly
species,
on individuals. So people seem to have populations that are probably very strongly related to their traditional diets that it's not very predictable. Certainly it's not true across all humans. So it seems to be fairly idiosyncratic, but people do tend to have dominant populations in their gut microbiomes, and how those evolve is a subject of really intense research right now.
Right. Could I ask another question?
Sure.
Yeah, and how about that predominance referring to all these factors involved, for example, the milling method and the size, the particle size of of the brands, or.
So I'd like to think my hypothesis is starting to evolve, that I think that a lot of what we see in the idiosyncrasy of different individuals has to do with Their habitual consumption, especially, I'd like to say of cereal brands, maybe that's too cereal centric, but certainly of fibers in general, but of brands in specific, and cereal derived fibers in specific. And that could be, if that's the case, then if you, for example, have a really high wheat diet, one thing that we've seen over and over again is that in our cultures on wheat, we see Prevotella pop up over and over and over again, kind of whether or not we see that person's microbiome had a lot of Prevotella to start. We see Prevotella oftentimes, especially on small particles and in soluble fibers from wheat brands, be very abundant, which suggests that this organism has some kind of competitive advantage for that particular carbohydrate. And so my thinking now is that perhaps these kinds of things compound over sequential days. So if you are eating, for example, your a whole grain wheat bread over and over and over again, I wonder whether or not you're sequentially feeding certain populations and making them more stable in the gut microbiome. Interestingly enough, though, what we have found is that when we do the sequential batch transfer experiments, microbiomes from very different people become much more similar very rapidly. Usually within 3 cultures, we can start to see that those cultures look pretty similar. And what that suggests is that the microbes that are doing this work seem to be present across all humans, but maybe are just not abundant enough for us to be detected all the time.
Okay. And yeah, I want to thank you, Professor Steve, for your nice presentation.
Thank you.
Thank you.
I'm happy to do it. Second question. The enzymes of probiotics and gut residents involved in obtaining bioactive compounds have already been identified. So this is a very, very intense area of research. I think this is going to be kind of the next frontier. No, we know it's very poorly ascribed, the enzymes to which their potential functions. So what that means is that the microbiota that are really, what genes are those genes the same that they use, those proteins that they synthesize, are they the same across all individuals? We have some early data that suggests that pretty fine differences in fiber structure select for organisms with different enzymes of specific glycoside hydrolase families, for example. And so it looks like those enzymes are not functionally equivalent, even though kind of at the bioinformatic level, we have sort of assumed that they're the same based on their similarity to one another. It seems like that's likely to be not the case. So this is a huge area of research. We're going to need a lot of time to figure out how microbes are modifying food, how they're gaining access to nutrients. And we're going to need to, as you mentioned, Miguel, get something where we have some very concrete mechanistic understanding of how these fibers are feeding certain microbes and causing them to bloom. Next question regarding why did we do 7 days? We picked 7 days from previous research where we had done the same thing with soluble fibers. So what we see usually with soluble fibers is that populations really stabilize after 3 to 4 individual day passages. Now, soluble fibers are easier to ferment than brands are, so you could make the argument that perhaps we should have gone a little further with that, but we did not. We took those off at 7 days for that reason, because we were seeing normalization of community structures in that time frame. Now, the particles and the attached particles seem to show that there still is a lot of donor-dependent behavior after 7 days of fermentation with bran particles, which is interesting to us. We did not use a static fermentation model. This is a slowly shaken fermentation model. There is a little bit of shear force. It's not a lot, but mostly it's enough to keep the particles suspended and allow opportunities for microbes to find them, attach to them, and consume them. I don't know if does that answer the question?
I have a question.
Please go ahead.
There's a problem with the sound. Yes, parezca, tal vez puedas escribir tu pregunta en el chat. Let's wait a little bit for Varesca. We are asking her to write her question. Probably works better.
No sé si me escuchan.
Yeah, Varesca.
Okay, thank you. Nice to meet you, Professor Liederman. I understand that microbiota abundance are related to the particle size and milling method of brands. But we can see that the same microbial behavior between donors is not the same. And I have two questions for you. Why do you use only 2 donors? And if the variability between donors improve if we increase the number of donors?
Yeah, that's a good question. So the answer to the question is when we started to do these kinds of sequential batch cultivation experiments, we had very little to go on with respect to how many donors it would take in order to get repeatable kinds of results across people. So we had our preliminary data was from soluble fibers, and like I said before, those soluble fibers really strongly recruit very similar communities across very different donor profiles, and they do so pretty rapidly. So that was the amount of deterministic behavior we see for those soluble fibers, like for example, even arabinoxylans solubilized from brands, we see very similar profiles. And I didn't present that data today, but we have one paper on that already. There's another one we'll have coming out very soon. So we use that as the benchmark for our particle sizes. And what we find is that the particles and the insoluble fibers in general are better at maintaining diversity than soluble fibers do. That makes sense. They're more diverse and there's more nutrients, different types of nutrients available to different microbes. But they also have idiosyncrasy, like we don't tend to see for our individual, for our soluble fibers. So I think, yes, we proposed 3 in the grant, so that's the way that we had started to do it initially. I think what needs to be done in the future now is that we need to maybe do less individual looking at the different kinds of maybe milling methods and the particle size interactions with these 3 donors. So now that we've seen some effects that are derived from that, maybe we need to pick 1 method or 2 methods or 3 methods in the same particle size or some kinds of things like that. And then we can branch out among the number of donors that we try on the next grant. I think that would be a really useful thing to do.
Thank you.
Hello, I have 2 questions. Do you hear me?
Yes, I can.
Okay. I understand that you use different milling methods during the research. So, well, I have 2 questions. The first is, what milling methods were used during the experiment? And the second, what do you think was the best method and why?
So initially our work was with the cyclone mill, like I said before. We then branched out into the roller mill, the hammer mill, and the stone mill approach to processing the brands. Honestly, with respect to the results that we're seeing for short chain fatty acid production, actually the roller mill seems like we're seeing better short chain fatty acid production from the roller milled brands than we are from the others. And so if that's our goal for success, which I think is the most reasonable goal for success right now, roller milled brands seem to do the best job. And specifically the smaller you can get the brands, the better. Now, I think we need to do more work in order to say that with some confidence, but that's where we're leaning right now.
Thank you for your interesting conference.
Thank you.
Steve, I had a question, Steve.
Yes, sir.
I understand that you use a brand, rice bran, etc., and to milling, but another is to fermentation, right?
Mm-hmm.
What happened when you prepare a food, for example, that include a dietary fiber ingredient to increase the dietary fiber in the food? Is the same criteria for to sample preparation when you had a food like a snack, a bakery product, you know?
Yeah, so I wish I could answer that question. We really want to do that experiment. We proposed it in our last submission to the USDA. We did not get funded on that one, so we have not had an opportunity to do that experiment yet. But the hope is that we'd like to try to see whether or not this is making any difference in people's gut microbiomes by, feeding them differently milled or differently sized particles. We were probably starting with kind of a bran muffin approach was our proposed work. But some diversity in diet would be a good idea to try to keep compliance high. But we would really like to see that experiment be done in the next year or so.
Okay, thank you. The other question is, I don't know what is the main component perhaps in the fermentation for the microbiota. Is a physical structure or is a chemical structure? When you pass in sequential pass in the milling, what do you think about is changing the structure of the undigestible carbohydrate present in the sample, or is a physical more than chemical change in the sample?
Yeah, so I should be clear. I don't think I explained it well. So when we are doing our sequential transfers, we're transferring the microbes, not the particles. So the idea there is that we are giving them fresh particles with each generation. So you're enriching for microbes that consume those particles over time. And so in this case, with the bran particles, I think it's the physical structure that's determining the microbial reactions more than the chemicals. But those could be dependent on the type of bran fiber. The physical kinds of interactions we see may be dependent upon the type of arabinoxylan structures, for example, that are presented to those microbes. We certainly have seen before in previous work that differences in arabinoxylan structure are very, very impactful on which microbes are able to survive, especially in sequential batch experiments. So I think, I can't tell you for a fact it's not the chemical structure of the fibers that's driving it, but it would be really, so what I would really like to be able to do is to look at the identical kinds of compositions with respect to arabinoxylan structure at the same particle size. And then that would, and that argues for doing things like Bruce Hamaker's lab has done, like making artificial particles using thin film deposition, for example, 3D printing artificial brands with arabinoxylans of different branchedness, as an example. That's probably where we're going to have to go in order to make kind of like a fake insoluble particle to try to test that hypothesis first, because it's really hard to separate those two variables.
Okay, thank you. I had the last question, Steve. Sorry.
Oh, that's great.
Recently, I read that a new classification of resistant starch. The resistant starch is part of the dietary fiber, and the new classification, the traditional classification of resistant starch is based in the environment and the chemical structure of the starch. What do you think about of the new classification? This is based on the type of bacterial species that produce resistant starch. The new classification suggests if the resistant starch is slow or rapidly fermentable by the microbiota, etc. do you think that it's possible to translate this to the dietary fiber to classification based on the products of the, as well, of the short chain fatty acids that produce the fiber? I don't know.
So I will say I'm no starch expert. So take everything that I say with a grain of salt, but I do think that we see differences that are imposed by starch structure in the experiments that we've done, where microbes are being selected by slightly different starch structures, both of them seemingly to be resistant starches. And so I think that starch structure at the macro level is going to be determinative for which microbes are able to grow well and which ones aren't. So we have this kind of, I think we have a too simple understanding of how starch structure interacts with the gut microbiota. I would really like to see some differentiation among the different starch types that is based upon things like performance in the gut microbiome. It's simple enough, right? So it's very expensive, but it's simple enough to do things like C13 label starches, and especially C13 labeling starches with different structures, and then looking at, essentially metabolism rate by humans could be really interesting. So for example, if we see production of CO2 from C13, a C13 containing CO2, after we see, passage of a meal through like the orocecal transit, we start to see that hydrogen peak that we get as soon as materials end up in the cecum or terminal ileum. That could be really interesting. Do starches, for example, are they all consumed, all of the resistant starches in the ascending colon, or do some of them make it to the transverse or even descending colon? It would be really, really important to know that. And I think that they probably perform very differently. So this is something I think that is very understudied, and I would really like to see more research develop towards that angle.
Okay, thank you. Any question for attend? No hay más preguntas en el chat. No sé si. Okay. Bueno,
I have a question.
Okay, Ariana, go ahead.
And Dr. Steve, my question is regarding the genera and the species that are found in the human gut. So are they similar to the microbiome found in other mammals? This is all new for me. It's a topic that I really know, I will say nothing. Yeah.
Yeah, so oftentimes you'll find related species across different species, like mammalian species. For example, there are microbiota, like Bacteroides species, that are specific to mice. They seem to do very well in mice. And what you'll find oftentimes is that if you try to engraft human microbiota into the mouse model, the mouse microbial species will rapidly outcompete the human ones. So it suggests that there are some specific adaptations to different host organisms, but they're not, I think, so firm that that species don't move across organisms. Like, for example, the dietary structure of pigs is much more similar to humans in the sense that we're both monogastrics. We can see some very similar species in swine microbiota, oftentimes not identical species, but similar species across humans and swine populations. So is it possible for there to be crosstalk or communication among those? Absolutely. Although we don't know what the efficiency of that is. I would imagine that if you get higher into primates, our microbiomes will get more similar, although I haven't really done a whole lot of work trying to study that.
Okay. Thank you very much. And the other question is, I think it's a very basic one. How a newborn gets like his or her gut microbiome? Mm-hmm. A human newborn.
That's interesting. Yeah, so it's interesting. We know a lot from birth mode studies that, infants that are born by cesarean section end up with a gut microbiome that much more strongly resembles skin, at least to start. And infants that are born vaginally have a microbiome that is much more similar to a normal, or rapidly develops into a much more similar gut microbiome. And so what it looks like is, from the infant, you're inoculated with whatever you encounter first. So that inoculation then determines at least the initial part of the succession. Now, you can start to see that those microbiome structures normalize over time, but then you're into selection by formula versus breast milk and various different nutrient environments. You can see those communities can change depending on how the life history of the infant. But mostly it looks like our initial inoculum is very, very early. Like as soon as we're born, whatever we encounter first has the advantage.
Okay, thank you very much.
Thank you very much, Steve. I will share my screen. Ok, sí, pueden ver mi pantalla, ¿no?
No, doctor.
A
No,
ver,
doctor.
no sé si, a ver. No, no se logra. No.
Esperamos un poquito más.
A ver, no sé. Creo que no puedo compartir la pantalla. No sé, Rosa Isela, si tú tengas el certificado. Y lo puedas compartir, Rosa Isela.
Sí, lo comparto.
Ah, gracias, muy amable. Gracias. Gracias. Steve, the authorities of the Center for Development of Bioproducts at Instituto Politécnico Nacional award the present certificate to Dr. Steve R. Lindemann for the excellent talk. Thank you very much, Steve. I will send the certificate by email. I appreciate your participation, your keenly, because always attend the emails. That is great for you. Thank you very much. I hope to visit West Lafayette soon, perhaps in May. I am planning.
Oh, great.
I will try to talk a little bit with you. Thank you.
Yes, please. Excellent. Thank you so much.
Thank you. Thank you very much. Gracias a todos los asistentes y bueno, continuamos para la siguiente semana, creo que es hasta el 14 de marzo. Entonces, no sé si Gaby, alguna cosa o Perla quisieran agregar. ¿No? Bueno, pues entonces les damos las gracias por su atención. Steve, muchas gracias, Steve. I know that you speak a little bit Spanish. It's nice. Thank you.
You're welcome. Thank you.
Bye-bye.
Buenas tardes.
Buenas tardes.